अस्सलाम वालेकुम माय डियर स्टूडेंट्स। आज का जो मेरा यहाँ पे लेक्चर है इट इज़ फॉर क्लास 11th एंड यू नो दैट द टॉपिक that we were discussing here, it is sexual reproduction in bacteria. I had told you that the sexual reproduction in bacteria take place by three methods. One is conjugation, another is transformation, and one more is transduction. Conjugation and transformation we have already discussed, and now today the topic that we are going to discuss here it is transduction. So my dear students, as far as transduction is concerned, it is a method of genetic recombination in bacteria in which a segment of DNA is transferred from one bacterial cell to another bacterial cell with the help of aberrant bacteriophages. मतलब ये कि ट्रांसडक्शन इज ऑल्सो a method of sexual reproduction or genetic recombination in which there is no direct contact between the two bacterial cells, but still there is the transfer of a segment of DNA that carries a number of genes from one bacterial cell to another bacterial cell without any direct contact in between the two bacterial cells. And the segment of DNA is transferred from one bacterial cell to another bacterial cell with the help of viruses called bacteriophages, and more appropriately we should say aberrant bacteriophages. तो अब आप ये याद रखोगे, कंजुगेशन में जब डीएनए सेगमेंट ट्रांसफर होता है इट इज ट्रांसफर्ड थ्रू कंजुगेशन ट्यूब और ब्रिज एंड देयर इज डायरेक्ट कॉन्टैक्ट इन बिटवीन द बैक्टीरियल सेल्स. तो जो ट्रांसफॉर्मेशन आपने पढ़ा, इन ट्रांसफॉर्मेशन देयर इज अ ट्रांसफर ऑफ सेगमेंट ऑफ डीएनए टू बैक्टीरियल सेल थ्रू द लिक्विड मीडियम. लेकिन यहाँ पे आपको याद रखना है, इन ट्रांसडक्शन these are the bacteriophages that act as the third party. They in fact mediate the transfer of segment of DNA from one bacterial cell to another bacterial cell. तो यहाँ पे आप याद रखिए, transduction was discovered by Zinder and Lederberg in Salmonella typhimurium. In this bacterium transduction was being discovered in 1952. तो यहाँ पे आप देखिए कि बैक्टीरियोफेजेस होते क्या है. बैक्टीरियोफेजेस और द स्पेशल वायरसेस दैट अटैक बैक्टीरियल सेल्स फॉर द पर्पस ऑफ देयर मल्टीप्लिकेशन. बैक्टीरियोफेज जो है दे वर डिस्कवर्ड इंडिपेंडेंटली बाय टू साइंटिस्ट्स. वन वाज टूवर्ट एंड अनदर वाज डे हैरिली इन 1915 and 17. So they discovered them independently and coined the term bacteriophages for such viruses. However, bacteriophage literally means a virus that eat upon the bacteria. लेकिन bacteriophages they don't need food. They are at the cross border between living and non-living things. तो bacteriophages in fact attack bacterial cells for the purpose that they have to replicate their DNA for their multiplication, and bacteriophages do not possess the machinery, enzyme machinery. They don't possess the energy for their DNA replication. So like viruses, bacteriophages are acellular. So you can see here they are of various shapes, but this is the most common shape of bacteriophages. And you can see like viruses, just like they are also viruses, these bacteriophages consist of, they are acellular, they consist of genetic material, and in them genetic material is usually DNA, and that genetic material is externally bounded by a proteinaceous covering, that is capsid. So structurally they consist of many parts. One is head, in which there is enclosure the genetic material, that's DNA. Then there is collar, and there is the tail, that's contractile. To the tail are attached tail fibers or spikes. So when the bacteriophages get attached to the bacterial cells, there is collision in between the bacteriophage with the bacterial cell. The bacteriophages possess some proteins called pilot proteins which are present at the tip of their spikes. By pilot proteins, they get attached to the bacterial cells, and in bacterial cells there are specific receptors in their cell wall to which these pilot proteins get attached, and this is called attachment or adsorption. And you have to remember, specific bacteriophage can attack specific type of bacterial cell. After the attachment, this bacteriophage, it penetrates its DNA inside the bacterial cell, and when the DNA is penetrated inside the bacterial cell, this viral DNA replicates to form hundreds of copies by using the energy and enzyme machinery of the host bacterial cell. When hundreds of copies of bacteriophage DNA are formed, each bacteriophage DNA gets assembled. It forms a new capsid and get assembled into new bacteriophage. And when hundreds of bacteriophages are formed in this host bacterial cell, ultimately they cause the lysis breakdown of the bacterial cell, cause its death, and the bacteriophage get escaped out. This is how bacteriophages complete their life cycle by the help of host bacterial cells. So my dear students, you have to remember here, how can you differentiate viral DNA, that is bacteriophage DNA, from the bacterial DNA of the host cell? You should remember DNA, as you are also knowing, DNA possesses the four types of nitrogenous bases. One is adenine, guanine, thymine, cytosine. And you have to remember that the viral DNA is characterized by the presence of modified form of cytosine called 5-hydroxymethylcytosine. तो once again I will repeat, तो वायरल डीएनए और बैक्टीरियल डीएनए डिफरेंशिएट होता है इस बेसिस पे। बैक्टीरियल डीएनए में होता है नॉर्मली साइटोसिन नाइट्रोजनस बेस, लेकिन यहाँ पे जो साइटोसिन होते हैं इन वायरल डीएनए दैट इज इन मॉडिफाइड फॉर्म कॉल्ड 5-hydroxymethyl cytosine, एंड दैट प्रोटेक्ट्स दिस वायरल डीएनए अगेंस्ट द अटैक ऑफ न्यूक्लियस एंजाइम्स। तो यहाँ पे देखिए हाउ ट्रांसडक्शन टेक्स प्लेस विद द हेल्प ऑफ बैक्टीरियोफेजेस। तो कभी कभार ऐसा भी होता है, समटाइम्स दिस हैपन व्हेन दिस bacterial DNA gets fragmentized into a number of fragments. Sometimes this may happen a segment of bacterial DNA, a segment of bacterial DNA, it recombines, it gets integrated with the viral DNA, bacteriophage DNA. When this bacteriophage DNA gets assembled to form a new bacteriophage, that new bacteriophage is termed as aberrant bacteriophage. तो अब आप याद रखिए, aberrant bacteriophage वो bacteriophage होता है in the DNA of which is integrated a segment of bacterial DNA from the host bacterial cell, and this aberrant bacteriophage carrying the segment of bacterial DNA, when it attack another bacterial cell of the same species like this bacterial cell, this aberrant bacteriophage is not able to cause the lysis of this bacterial cell. It does not damage it, and in fact it benefits this bacterial cell. When it penetrates it is viral DNA, that segment of bacterial DNA that it has carried from this host bacterial cell, that gets integrated, recombines with the DNA of this bacterial cell and bring genetic recombination in this bacterial cell. So this bacterial cell has got benefited. So it has undergone genetic recombination, it will experience the variations, and it will fulfill the purpose like other organisms fulfill by performing the sexual reproduction. So we can say simply a segment of DNA has been transferred from one bacterial cell to another bacterial cell with the help of bacteriophage, aberrant bacteriophage. This aberrant bacteriophage has acted as a third party, and this is the transduction has taken place. and a genetic recombination has been brought in this bacterial cell without undergoing lysis, as aberrant bacteriophages lost the ability to cause the lysis of bacterial cells. So कल आपको कोई ये पूछेगा, if bacteriophages transfer the segment of DNA from one bacterial cell to another bacterial cell, जिस bacterial cell में वो segment of DNA transfer करेंगे तो definitely they will cause its lysis as well. So what is the fun of undergoing genetic recombination in this bacterial cell? तो उस वक्त आप ये कहोगे the bacteriophages are aberrant. They already have lost the ability to cause the lysis of bacterial cells. Okay, now is here. Bacteriophages are of two types. One are called as lytic phages. For example, T2 phages, T4 phages are the examples of lytic phages. Another type of bacteriophages are called lysogenic phages. They are also called temperate phages. For example, lambda phages. These are the example of lysogenic phages. तो देखिए यहाँ पे first of all lytic phages क्या होते हैं. Lytic phages are the type of bacteriophages. When they attack the bacterial cell, they cause it is immediate lysis, its immediate breakdown and result in the death of the host bacterial cell. तो यहाँ पे आप देखिए क्या होता है. This is the lytic phage. It has got attached to the host bacterial cell with the help of its pilot proteins at the tip of spikes. So when this lytic phage penetrates its viral DNA inside the host bacterial cell, it fragmentizes the bacterial DNA into a number of segments. And normally क्या होता है, this viral DNA replicates to form hundreds of copies, and those hundreds of viral DNA copies get assembled into new lytic phages. Those lytic phages ultimately cause the death or lysis of host bacterial cell and escape out. But sometimes this may happen, कभी ऐसा भी होता है, this is the viral DNA of lytic phage, and any segment of bacterial DNA, randomly recombines, integrates with the viral DNA, and when this viral DNA get assembled, when this phage DNA get assembled to form a new lytic phage, that lytic phage is aberrant phage, as it has carried a segment of DNA from the host bacterial cell. So any random segment of bacterial DNA has got incorporated with phage DNA, and this aberrant lytic phage, when it will attack another bacterial cell of the same species like this bacterial cell, and it will penetrate its viral DNA that carries the segment of DNA from this bacterial cell. तो क्या होता है यहाँ पे, this aberrant lytic phage does not cause the lysis of this host bacterial cell. In fact, it will benefit it. The segment of bacterial DNA it has carried from the previous bacterial cell, that will recombine with the DNA of next or another host bacterial cell, and that segment of DNA will bring genetic recombination in this bacterial cell. So you can say a segment of DNA has been transferred from this bacterial cell to this bacterial cell, and this bacterial cell has undergone genetic recombination. It will experience the variation, so we can say transduction has taken place, but this type of transduction is called as generalized transduction. तो अब आप ये देखिए, what is generalized transduction? It is the type of transduction in which any random segment of DNA is transferred from one bacterial cell to another bacterial cell. Means there is any segment of DNA that can get transferred from one bacterial cell to another bacterial cell, and this is brought about by lytic phages. Okay. Now lysogenic phages, also called temperate phages, like lambda phages. These are the type of bacteriophages. When they attack the bacterial cells, they don't cause their immediate lysis. You have to remember like this. So this is a lysogenic phage. When it gets attached to the bacterial cell with the help of pilot proteins present at its spikes, it penetrates its viral DNA inside the host bacterial cell through contractile tail. And what happens here? The viral DNA gets integrated, it recombines with the DNA of host bacterial cell in inactivated form. तो यहाँ पे क्या हुआ? There has not been fragmentized the host bacterial DNA, it's intact. यहाँ पे जो viral DNA है lysogenic phage का, it has recombined with the DNA of the host bacterial cell in inactivated form. Basically in the viral DNA there are present some genes called inhibitor genes. Those inhibitor genes code for the proteins called repressor proteins, and these repressor proteins keep this viral DNA in inactivated form, and this viral DNA is termed here as prophage. So this bacterial cell, it is said to be lysogenic. So what is a lysogenic bacterial cell? It is such a bacterial cell that possess viral DNA of lysogenic phage integrated in its nuclear DNA in inactivated form. So this lysogenic bacterial cell, without showing any symptoms, it will divide by binary fission. One bacterial cell will divide into two bacterial cells, two bacterial cells will divide into four bacterial cells, 4 into 8, 8 into 16, 16 into 32. So the bacterial cell that's lysogenic, it will increase its number geometrically. It will survive, and those next generations of this bacterial cell, those are all lysogenic, they will survive for a number of generations, for 2 years, 3, 4 years, 5 years. So sometimes this may happen. This lyso among a number of lysogenic bacterial cell, due to some circumstances, due to some conditions, like when such bacterial cells are exposed to X-rays, to UV rays, they are exposed to concentrated heavy chemicals, or they are exposed to high temperature, these this viral DNA that gets activated, the repressor proteins are damaged by those conditions. With the result, this viral DNA in this lysogenic bacterial cell gets activated, and this viral DNA gets detached from the host bacterial DNA and replicate to form a number of copies, hundreds of copies. And क्या होता है यहाँ पे? Each viral DNA of lysogenic bacterial of lysogenic phage that ultimately get assembled to form new lysogenic phages, and those lysogenic phages then cause the lysis of one of the lysogenic bacterial cell and get escaped. लेकिन यहाँ पे देखिए what is the point here. Sometimes this may happen, कभी ऐसा भी होता है. This is a lysogenic bacterial cell, and here the viral DNA of lysogenic phage, it has got activated. तो जब ये viral DNA activate होता है due to some circumstances, this viral DNA gets detached. ये viral DNA यहाँ पे detach होता है from the host bacterial cell. तो जब ये detach होता है, it may carry with it a segment of DNA, either this segment or this segment of DNA. So the viral DNA, when it gets detached from the host bacterial cell, it carries a segment of DNA, either this segment of DNA or this segment of DNA, not these segments, this or this. So we can say when it will carry with it a segment of DNA, here we can say a specific segment of DNA or bacterial DNA has got incorporated with the viral DNA. When this viral DNA will get assembled to form a new lysogenic phage, that's also aberrant phage. This is here a new bacteriophage that's lysogenic, carrying a specific segment of bacterial DNA incorporated in its DNA, that's aberrant phage. When this will attack another bacterial cell of the same species like this bacterial cell, so this aberrant phage, that is a lysogenic phage carrying a specific segment of bacterial DNA from this bacterial cell, when it will penetrate its viral DNA inside the bacterial cell, this specific segment of bacterial DNA will get integrated with the bacterial DNA of this bacterial cell, next bacterial cell, and with the result it will bring genetic recombination in this bacterial cell. तो यहाँ पे भी क्या हुआ, a segment of DNA has been transferred, but a specific segment of DNA has been transferred from one bacterial cell to another bacterial cell, and this is also transduction called as specialized transduction. तो यहाँ पे आप याद रखिए जो specialized transduction है, what is it? It is the type of transduction in which a specific segment of DNA is transferred from one bacterial cell to another bacterial cell, and this type of transduction is being carried by lysogenic phages. तो अब आपको ये यहाँ पे अंडरस्टैंड हुआ, in transduction there is transferred a segment of DNA from one bacterial cell to another bacterial cell with the help of bacteriophages called aberrant phages, and these bacteriophages act as third party in transduction. But transduction is of two types, one is called generalized and specialized transduction, and I hope that you might have understood this well, that what's transduction, what's generalized and specialized transduction. And very importantly, you have to remember that generalized transduction is being carried by lytic phages, whereas specialized transduction is being carried by lysogenic phages, also called temperate phages. तो यहाँ पे आपको ये क्लियर होना चाहिए कि आप कह रहे होंगे कि ये स्पेसिफिक सेगमेंट ऑफ डीएनए क्या हुआ, मतलब अगर आप यहाँ पे कहोगे देयर आर मेनी सेगमेंट्स इन बैक्टीरियल डीएनए, सेगमेंट फर्स्ट, सेकंड, थर्ड, फोर्थ एंड फिफ्थ, तो यहाँ पे कौन सा सेगमेंट इंटीग्रेट हो सकता है विद वायरल डीएनए, आइदर द सेगमेंट फर्स्ट और सेगमेंट फिफ्थ, दीज सेगमेंट्स कांट गेट इंटीग्रेटेड. So that's why we say here a specific segment of bacterial DNA has been transferred to next another bacterial cell by lysogenic phage, which is called specialized transduction. Okay, so that's all about the transduction. Thank you very much.